My astounding pleasure to herald Dr. Syamdas Bandyopadhyay as studio speaker. Dr. Bandyopadhyay furnished his MSc from Benares Hindu University and achieved his PhD in physiology in working at National Center for Cell Science Pune, being registered at Vidyasagar University, Midnapur, West Bengal, under the joint supervision of Dr. Bhaskar Saha of NCCS Pune and Professor Somnath Roy of Vidyasagar University. He has also the experience to work with some eminent scientists like Dr. M.V. Krishna Shastri, Dr. Devashish Mitra, and Dr. Shailaja Singh of NCCS Pune. From 2013 to 2016, he remained research assistant at ICMR Kolkata and research associate at Bose Institute of Kolkata. From 2016 to 2019, he acted as postdoctoral research associate in Yale School of Medicine, Yale University, USA, and associate research scientist of the same university, respectively. He has 9 highly acclaimed international publications and having the experience of either attending or delivering lectures or presenting posters in 5 seminars in India and abroad, as well as experience in reviewing scientific articles in Elsevier editorial system. He is also a member of American Association of Immunologists. Now, with this view introduction, I request Dr. Bandyopadhyay to deliver his lecture. Thank you.
Shall I start?
Yes, you
Okay.
start.
Thank you very much, sir, for such a nice introduction. I think this seminar is going to be for the students. So today I'm going to speak about the immunoassays. Those are RIA and ELISA. So I think these are the two assays that is there in your syllabus of CC10 in immunology of semester 4, as well as in semester 6 TSC4 in endocrinology paper. So if you see that you have these two topics of immunoassays, ELISA and RIA, as chapter 3 in unit 4, right, as well as here in chapter 2 in unit 4 of semester 6. So first, let us understand, before we go into the details of the ELISA and RIA, let us first understand the basics of the interaction between antigen and antibody interactions. Understanding of this interaction is important because the ELISA or RIA, the principle is based on the antigen and antibody interactions. So this one is the surface of an antibody that's going to interact with the surface of antigen. However, this interaction doesn't happen with the whole surface of an antigen, right? So as you know, the interaction happens between a small part of the antigen, which serves as the antigenic determinant called epitope, with a smaller part of the antibody, right? So these two portions interact with each other involving various non-covalent interactions. For example, hydrogen bond, then ionic bond, you know, hydrophilic interactions, van der Waals interactions, ionic bonds, and all these are bonds they contribute in forming the non-covalent interaction between epitope with antibodies. So now if we would like to first define what is affinity. So affinity, it is the combined strength of the non-covalent interactions between a single antigen binding site, that means one epitope on an antibody, and a single epitope and a single, this is called paratope, right? That's the present on the antigen binding site. So it is the interaction 1:1, and the combined strength of the interaction is called the affinity. Now if you see the chemistry of the interaction, so here the antigen that tends to combine with antibody, and that forms the antigen antibody complex, right? So now if you see, the K1 is the forward rate constant, and K-1 is the reverse rate constant, as has been given conventionally. So now if you would like to calculate the association constant, the Ka association constant is the Ka, that is the ratio of K1 to that of K-1, right? And now if you put the values, the concentration values of the K1, it becomes antigen antibody bound complex to the ratio of free antibody and antigen complex, right? So this give you the value of the association constant. So you have to first understand that this is the way the epitope of an antigen interacts with a surface of antibody. So with this understanding, now we'll directly go to what is radioimmunoassay, right? So as the name indicates, radioimmunoassay, it involves some kind of radioactivity, right? So the two scientists, Varsan and Yalo, in 1960, they first measured the endogenous plasma insulin using this technique. And this led them to receive Nobel Prize in 1977. So Yalo received Nobel Prize for this major milestone discovery in immunology. So the principle of radioimmunoassay is based on the competitive binding of radio-labeled antigen. So here you have a radio-labeled antigen. We call it radio-labeled because this antigen has some kind of radioactivity. So radioactivity has been introduced, and that is why this is labeled, right? And that competes with the same antigen, which is unlabeled, right? So this is the antigen that you would like to measure the concentration of this particular antigen. Now you can ask me the question, where from this antigen comes? So this antigen may come from serum, it may come from any component of the cell, inside the cell, or even outside the cell, right? So sometimes the cells secrete cytokines or any kind of molecules, concentration of which can be measured using the radioimmunoassay. As I said, I repeat, the radioimmunoassay is based on the principle of competitive binding between an labeled antigen with an unlabeled antigen. So they compete with each other, and then you can measure the radioactivity. That will give you an indication of, for example, how much antigen is present in the serum. So your aim here is to estimate the concentration of the antigen that is present in the serum. Let's say you have done an experiment. So you have injected certain molecule in a mouse, and then you would like to measure the concentration of one cytokine, let's say IL-10 or IL-12. Now this is the antigen, IL-10 or IL-12. So you would like to measure the concentration of this IL-10 or IL-12 by using this kind of competition, right? So this is called radioimmunoassay. And as you see, because the radioactivity is involved, so it is called radioimmunoassay. Now notice that here, this is the infected sample, right? So that means this is your experimental sample. Now you need to have a control. What is the control here? Here the control is that you have to have a mouse that is not injected with any kind of antigen from outside. So this is the serum that we collect from that mouse is called uninfected serum. So here you don't expect this kind of antigen, right? So you have to see the value of uninfected control as well. So most probably you will get very little signal for uninfected control, and you will get a lot of signal for infected control if the antigen stimulates the production of this kind of cytokine. So this is the basis of the radioimmunoassay. Now, what are the steps that are involved in radioimmunoassay? So there are multiple steps. I will not go into the great details, because that's not neither we have time nor it's required. So you just have to know the principal steps which are involved in performing radioimmunoassay. The first step, as you can see, it is to add the buffers to the tubes, right? So you just add some appropriate buffers. In step 2, you have to add known amounts of unlabeled antigen to the mixture. For the ease of understanding, let's assume this is IL-10, a cytokine that serves here as an antigen. And then this is supposed to compete for the binding sites of the antibodies, which is yet to be added in the sample. And in step 3, you add the radioactive antigens to the mixture. So same antigen, right? Here you are doing this radioimmunoassay for IL-10. You add IL-10, which is radioactively labeled in this step, right? Is it clear? And in step 4, you now add the antibody, right? So this antibody will tend to bind with this IL-10, both labeled as well as unlabeled. And in step 5, they will compete for binding. Now the antibody will not distinguish between labeled and unlabeled antigen, and therefore it will bind to all of these antigens. It will try to bind with all of these antigens. And if you have more unlabeled antigen, this will replace the radioactivity labeled antigen here for binding, right? So this radioactivity can be now measured using a proper system. So there are several systems available. One is called scintillation counter chamber. So you can now hold this test tube in the counting chamber, and then you can calculate the radioactivity. So now if you perform this step 2 with several known concentrations, and then finally if you get the value of the radioactivity at this step and plot these several values in a curve like this, then perhaps you'll get a kind of this kind of curve, right? So here you have used 1, 2, 3, 4, 5, 6, 7 known concentrations of the cytokines, right? So that makes the standard curve. So a standard curve can be constructed by plotting the percentage of antibody bound radiolabeled antigen against known concentrations of a standardized unlabeled antigen. So here you have taken 1, 2, 3, 4, 5, 6, 7 known concentrations of unlabeled antigen. So this curve allows determining the unknown antigen concentration directly from the standard curve. So this is, so far we described, is the construction of the standard curve using RIA or radioimmunoassay. Now if you have your sample of which you do not know the concentration of that antigen present in the sample, that you just simply put in this curve, and then you can calculate the value, right? So that will give you the kind of exact concentration that is present in the sample. So this basically, this is how you can, using radioimmunoassay, you can measure the concentration of a particular cytokine present in the sample or the serum, right? Okay, now what are the applications and limitations? So the applications is, right, so the test can be used to determine very small quantities of antigen. So even if it is present in picomolar or nanomolar concentrations, you can easily measure the concentrations of that antigen present in the sample. So that means it is highly sensitive. So this reaction is highly sensitive, which is required because sometime in experiment you get very little amount of cytokines, and that you want to measure, right? So in that case, RIA will be very much suitable as a technique to be used. And then next, the specificity. The test is used for quantitation of hormones, drugs, and antigens and other viral antigens. So that is what is called specificity. So that means this test is very much specific for a particular antigen. So you can avoid the cross reaction, and therefore you can make it more specific, right? So those are the principal applications of radioimmunoassay. Now you have certain limitations also. There are certain limitations of radioimmunoassay. So for example, the first is the cost of the equipment and reagents. So that means you have to spend a lot of money in order to set up such kind of system in your laboratory. And then you have the shorter half-life or of the radio level compounds. So that means if you purchase these radio level antigens in your laboratory, so because they have very shorter half-life, so they will decay after some time. So that adds to the recurrent expenditure to your lab, right? And then the requirement of special counter of radioisotopes. So if you would like to measure the radioactivity, you have to have a scintillation counting chamber. So that means that you have to purchase a scintillation counting chamber, which perhaps costs lakhs of rupees. And then the problem is associated with the disposal of the radioactive waste. So here, so the radioactive waste, you'll always have to think for a protocol or a system that is friendly to the environment. Just because you're using the radioactive waste, so you'll have to think for a safe disposal of the radioactive waste, because if it mixes up with the environment, then it's a problem. It's a great, it's a serious problem to the health of our Mother Earth. Right? So you can, all right, so if you have any questions, you can always ask me. And next, so these are all about RIA, radioimmunoassay. Now we'll move to the ELISA, which is enzyme linked immunosorbent assay. So the full form of the ELISA is the enzyme linked immunosorbent assay. Now if you have understood RIA, then understanding ELISA is very easy, because the basic principle lies the same. So what only differs is that their detection system. So what happens is that ELISA is used as a diagnostic tools in medicine and as quality control measures in various industries. So if you have purified a novel protein or new protein, if you like to measure the concentration of the protein, or if you are doing some kind of experiments in which you are expecting a particular cytokine to be induced, then you can use ELISA. It's a very easy technique to measure the concentration of that antigen or the cytokines present in the sample. And ELISA is used as an analytical tools in biomedical research for the detection and quantification of specific antigens or antibodies in a given sample. So ELISA is an analytical tool. So you can always analyze the quantity of a particular antigen present in the sample. Now the good thing is ELISA is derived from RIA, radioimmunoassay, that we just discussed previously. Now what is ELISA is doing? Number one, ELISA is a biochemical assay that uses antibodies and enzyme mediated color change to detect the presence of either antigen or antibody in a given sample. So the principal thing involved in ELISA is an enzyme mediated color change. So there will be a role of enzyme somewhere in the protocol that is associated with change of the color, and that you measure that change of the intensity of the color, and that will give you an indication of how much antigen is present in the sample. So ELISA, as the name suggests, it is an technique that involves a certain enzyme associated with change of the color. Now number two, both indirect and sandwich ELISAs allow detection of antigen or antibody at very low concentrations. So we have not discussed the indirect and sandwich ELISA till now. So we'll touch upon those techniques, I mean the variations of those techniques of ELISA subsequently. The competitive method detects compositional differences. So the competitive method is another variation of ELISA that will detect the compositional differences. That means if a particular antigen is made up of one or made up of two or more subunits, so you can always measure the differences, those differences present in their structure. So that too with very high sensitivity, even when the specific detecting antibody is present in relatively small amount, right? Okay, now ELISA is also not without limitations. It also has certain kind of limitations. For example, the enzyme mediated color change will react indefinitely. So when this color change happens, it can react without, it can react indefinitely. So it will not stop, right? So that means after over a sufficiently long period of time, the color strength will inaccurately reflect the amount of primary antibody present. So the color change will keeps on happening. Therefore, after certain time, the color may saturate also. So the amount of color or the intensity of color that you see in the sample may not reflect the actual concentration of the antigen. So that is one limitation of the ELISA. However, with the advent of the modern techniques, we have been able to overcome those limitations. So you can always stop the reaction using certain chemicals, right? So you can stop the reaction and you can make the intensity fixed at that particular time, which you can easily measure using a detection system. And number two, to detect a given antibody or antigen, a known reciprocal antigen or antibody must be generated. I mean, this is really funny because, ELISA is a technique that involves antigen-antibody interactions. So you always have to have a pair of antigen and antibody pair, right? So that means what essentially this point suggests you is that you have to purify the corresponding antibody. So even if this antibody is not available in the market, you have to make it for using one or other technique. So that means you must make the antigen-antibody pair available in order to perform enzyme-linked immunosorbent assay or ELISA. And three, the non-specific binding of the antibody or antigen to the plate will lead to a falsely high positive result. So what is a false high positive result? That means sometimes the antibody that you are using, they can bind non-specifically to the wells, right? So if it binds non-specifically to the wall of the wells, then you'll get more intensity of the color, but that doesn't reflect the actual concentration of the antigen present in the sample. So this is called false positive result. So that is another problem with ELISA. So we always have some kind of measures to avoid that. You can perform more washing, and that will help you remove the non-specific interaction of the antibody. Now, how and why was ELISA developed, right? Before the advent of the ELISA, the radioimmunoassay employing radioactively labeled antigens and antibodies were used. So where the radioactivity served as the receptor signal indicating specific antigen or antibody. So, but as you know, that the radioimmunoassay posed a significant health risks to the researchers. So the alternatives were very much accepted, because you are using radioactivity, right? So that might harm the experimenter. I mean, somebody who does the experiment, and also it might cause significantly negative effect to the Mother Earth, because if you do not safely dispose the radioactive waste, then it's a problem. So together they imposed a significant health risk. So for that, the scientists were looking for a good alternative that will overcome the radioactivity problem of the RIA, and therefore people started looking for the alternative, and ELISA came up subsequently. So ELISA is a modification of the RIA. So now who are the scientific contributors? In 1960, the enzyme linking process was developed by two different teams, superheaded by Stratis Abramias and G.B. Pierce. So these are two great scientists who developed an assay to conjugate an enzyme with antibody, right? So Pierce is a great scientist, so who later on opened his own biotechnology company. In the same period, immunosorbent preparation technique was published by White and Porath. So together they published immunosorbent technique. And independent research papers were also published in 1971 by Peter Perlman and Ivan Ingvall at Stockholm University in Sweden. And then Anton Scootz and Bocke van Wiemen in the Netherlands produced the knowledge that go into making ELISA. So, I mean, the bottom line is that, so there are contributions of lots of scientists who helped developing the ELISA as a technique, right? So these are the name of the some scientists who helped this, who helped in developing the ELISA technique. Now, technically, there are multiple variations of ELISA. Sometimes even enzyme linking is not done. There are other methods for detection, but just because the principle lies the same, so although the newer assays use reporters that are not enzymes in most of the cases, the underlying principle of the assays are similar, as I'm saying. Therefore, these assays, then that means the modifications of the ELISA are also grouped as ELISA, right? They are also called ELISA. Now, let's come to what is the principle of ELISA. Now, ELISA, first you have to understand it's a multi-step process like RIA. So I can tell you in greater details, because ELISA is a very common technique performed in immunological laboratory and various biotechnology laboratory. So what are the steps of ELISA? I mean, that will tell you about the principle of ELISA, right? So the first step is the coating. Coating of antigen is adsorbed. So the antigens are adsorbed onto well in ELISA plate in coating buffer. This is an well, right? So this is a single well to which you are adding this antigen, correct? So in the antigen which is suspended in the buffer. So this buffer must be appropriate so that antigen does not degrade. Rather it helps in keeping its structural conformation so that it can get absorbed onto the walls of these wells. Now the next step, so the first step is the coating of the antigen. Sometimes we coat the antibody as well, depending upon which kind of ELISA you are using. And the next step is called blocking. So the blocking, in this step, a buffer containing unrelated protein is used to block the free sites in the wells. So you used an unrelated protein. That means sometimes, most of the time, people use BSA, bovine serum albumin, that contains the albumin protein that can block the surface of the walls where the antigens are not able to absorb. That means this actually reduces the non-specific binding. So remember, blocking is done to reduce the non-specific binding of antibody that we are going to use in the next step. Now, the next step is the detection, right? Here, the enzyme conjugated detection antibody binds the antigen. So This one is the enzyme conjugated. As if you can see this, there is an conjugation with this antibody, right? This conjugated thing is nothing but this is an enzyme. So that is tagged with an antibody. Now this antibody is able to recognize the antigen, and then you can check the readout. So as you apply a substrate, a specific substrate, which will be converted into a product. So the catalyzation is done by this particular enzyme, which is tagged with this antibody, right? So that means a substrate is converted into product, and that is associated with the color change. You see here, there was light blue color, that changes into a light yellow color. So this color change is associated with this conversion of substrate into product, right? So now what you can do, you can easily measure the intensity of the color using a spectrophotometer or using a multiplex reader. So that will tell you how much color has been generated. And this generation of this color is proportionate with the amount of antigen that is present in the sample when you added it first, right? So that means more antigens are present, more will be the intensity of the color. Less antigen is present, less will be the intensity of the color. So if you measure the intensity of the color using an appropriate system, so that will tell you how much antigen was present in the sample. So that's your aim. So these are the main principles of ELISA that involves this technique. Now, I mean, people, because of their requirements, they started thinking of various modifications of the ELISA. So the first step in an ELISA experiment is the immobilization of the antigen in a sample to the wall of the wells of a microtiter plate. So this can be achieved by direct absorption to the plate surface or by using a capture antibody. So depending upon these modifications, there are several types of ELISA. For example, after immobilization, a detection antibody is added, which binds to the adsorbed antigen, thereby leading to the formation of an antigen-antibody complex. In general, ELISAs can be grouped into 4 main categories, right? So the modifications of ELISA can be grouped into 4 different categories. For example, the one is called direct ELISA. The number 2 is called indirect ELISA. Then number 3, which is most commonly used in the biological laboratory or the immunological laboratory, is called sandwich ELISA. And then number 4 is called competitive ELISA. So these are the 4 principal modifications of the ELISA. Besides that, there are other modifications of ELISA. For example, people also do multiplex ELISA and other things, but we're not going to discuss those things. So let us briefly understand what are the different modifications of ELISA. So this one is the direct ELISA. So as you can see, there is a microtiter plate. So this is a well of a microtiter plate to which you have added kind of buffer, and then an antigen has been immobilized in the well of an ELISA plate. So this one is the antigen, the green color. So let's, for the ease of understanding, let us assume that this antigen is IL-12, a cytokine, right? So now, in the second step, in direct ELISA, what people do is that the antigen is then added with antibody, which is conjugated with a particular enzyme, right? So that is performed just by mixing this antibody in this buffer. So the concentration of antibody, a fixed concentration of antibody must be used. So what happens is that this antibody can interact with the epitope of this antigen, right? So in the next step, what you do is that, which has not been shown here, is that you add a kind of specific substrate of this enzyme. So if you add the substrate, what happens is that there will be a catalyzation of substrate into a product, right? And now assume a situation where the subject changes, the substrate changes into product associated with a particular color. So you see the color has been changed. Here it was light blue in color, and the color has been changed into light yellow, right? So now this yellow change, color change, intensity of the color change can be measured using a microplate reader, and the value of that particular color will tell you how much antigen was present. So amount of color present is directly proportional to the amount of antigen present in the sample. All right. So now you also have, as I said earlier, like you also have a way to stop this reaction. So you sometimes we add a stop solution so that the color no longer changes, it is even fixed. So in that way it helps you to saturate the color, right? Okay. Now there are certain advantages as well as disadvantages of performing direct ELISA. For example, advantages are it is faster than any other types of ELISA. So you can perform this experiment in a couple of hours, right? That's the first advantage, very fast. And then the second advantage is that this technique has fewer steps. So these techniques involve only 2 or 3 steps. So that means less step means less number of errors. That's the third point. Less prone to errors just because it has very minimum number of steps. And then the fourth is the fewer reagents is required because minimum number of steps, so you need fewer number of reagents. So fewer number of reagents means less amount of money you spend for the experiment. So because in laboratory you always have to think for the amount of money that comes, right? Because government gives you the money to perform research. So you have to design the assay in a cost-effective manner so that you can perform maximum research using minimum amount of money. So that's a very important part of the experiment. Now there are also certain disadvantages associated with the direct ELISA. For example, the antigen immobilization is not specific. Sometimes they can bind non-specifically. Number 2, this may cause higher background noise than indirect ELISA. So higher background noise, right? So I mean, higher background noise means in control sample also you'll get a lots of color developed. So in that case, you cannot definitely come up with a conclusion. Number 3, less flexible. So each target protein needs a specific conjugated primary antibody. So that means this one is the primary antibody. So each primary antibody has to undergo a specific conjugation, right? And that makes the things more expensive if you want to conjugate every primary antibody. So that means it is less flexible. And then number 4 is the no signal amplification that reduces assay sensitivity. Here, because only one antigen is conjugated, is bound with one antibody, therefore there is no amplification of the signal, right? So that means that reduces the sensitivity. More amplification means more color. That means highly sensitive, but that's not the case here. Now we'll come to the next variation of the ELISA, which is called indirect ELISA. Here you see you have taken an antigen that is adsorbed onto the well of the ELISA plate, and then an unlabeled primary antibody binds to the specific antigen. So this is the unlabeled primary antibody that binds to the specific antigen. So this antibody is very much specific for this antigen. And in the next step, what you do is that you use a secondary antibody. So the blue colored antibody is called the secondary antibody that is tagged with a specific enzyme. And in the final step, what you do is that you add the substrate, right? This substrate can be catalyzed into a product because the secondary antibody is conjugated with a particular enzyme, and this substrate can now be converted into a product, and that conversion is associated with a particular color, right? So you see here is the light blue color has been changed into light yellow color. So the intensity of the color is directly again proportional to the amount of antigen that is absorbed in the first place. So that means more color is developed. That indicates that you had more antigen present in the sample. So now you can again use the multiplet reader, and using that you can determine how much color intensity of the of that sample or the well, and that intensity of the color will tell you how much antigen was present. Now there are certain advantages of performing indirect ELISA. So it is highly sensitive. Number 2, more than one labeled and labeled secondary antibody can bind to the primary antibody. So here you can just make, for example, one conjugated secondary antibody and use it for multiple primary antibody, because your antibody may be specific to the FC region, right? If you know the basic principles of the immunology, this region can be specific for the FC region, and therefore you don't need to perform the conjugation of every primary antibody, right? So that reduces the expenditure. So therefore it is economical. So fewer label antibodies are required. Then the fourth point is the greater flexibility. Different primary antibodies can be used with a single label secondary antibody. That is what I just described. So you have a great flexibility. And then the signal amplification. So here you can you can amplify the signal. That adds up to the advantages of using indirect ELISA. Now there are several disadvantages as well. So for example, the first is the possibility of background noise. So because you are using a secondary antibody pair, primary with secondary antibody pair, so this secondary antibody can non-specifically bind to the wall of the wells, and that can create a non-specific background noise. Then you have the second disadvantage, which is the secondary antibody may be cross-reactive. So cross-reactive means it can bind somewhere else, right? So that means it is not specific sometimes. So that adds up to the disadvantages of indirect ELISA. And three, the longer procedure than direct ELISA protocol, because more number of steps means much more time you require to perform this experiment. So that makes the protocol a bit longer, although it is best for determining total antibody concentration present in the sample. Now we'll come to the sandwich ELISA. So as you know, the sandwich, so you perhaps you all know about the sandwich, right? So here what you do in the first step, you first use a capture antibody and let it absorb onto the walls of this microreactor plate well, right? Individual well. So this is your capture antibody. We call it capture antibody. Then you add the sample in which you are expecting your antigen to be present. So this capture antibody, if it is specific for this antigen, this capture antibody will bind with this antigen. And in the next, or step 3, what happens is that this secondary, you use an antibody which will bind a different epitope of the same antigen, right? So this is called a detection antibody. So the first you use capture antibody, then you add the antigen or the sample serum, and then you add the detection antibody. The detection antibody is conjugated with a particular enzyme. So now if you, I mean, the final step is the same, just use the specific amount of substrate, add to this well, and that substrate will be converted or catalyzed into a colored product. And now you can calculate, you can measure the intensity of the color, and that intensity of the color will tell you how much antigen was present in your sample. That is the aim of the sandwich ELISA, right? So sandwich ELISA is one of the most used techniques in molecular biology or immunology laboratory. So because it has great advantages of high sensitivity, high specificity, it is very flexible, and fewer reagents are required, right? And although it has certain disadvantages also, like the antibody optimization can be difficult. What is optimization? Because your antibodies must be a pair which recognizes different epitope of the same antigen, right? So for example, this detection antibody, it recognizes an epitope that is different than the epitope recognized by the capture antibody. So in that way, you have to perform a little bit of optimization, right? So that is called standardization. Then number 3 disadvantage is that it needs a standardized ELISA kit or tested antibody pair. Of course, you have to perform various experiments in order to standardize the protocol before you perform the actual test, right? In case of sandwich ELISA. But it is best for analysis of complex samples, since the antigen doesn't need to be purified prior to the measurement. So in this case, you don't have to purify the antigen, just use the serum. You can add the serum and the antibody will automatically bind to the capture antibody. So it is best for that kind of analysis. And then we have the competition inhibition blocking ELISA. So this is the most complex type of ELISA. That is not, I mean, this is not routinely done in laboratory. This is for some specific uses. Here also you first perform the coating, and then you perform the blocking, and then you add the sample. So here you add the test sample mix to the wells, right? And then you add the detection antibody, and then you check the readout, intensity of the color which is developed. So competition or inhibition, also called blocking ELISA, also has certain advantages and disadvantages as has been enlisted here. I'm not going to the details of that because this is not routinely done in the laboratory, but I mean this is done for some specific uses. Here also you use a specific substrate that is converted into a colored product in presence of that conjugated enzyme that contains the enzyme, right? Here the substrate is called TMB substrate. Now, this slide shows you different modifications of the ELISA and summarize the concept. So, the concept that we received today is that the indirect ELISA, in which the antigens are coated first onto the well, and then you wash it to avoid the non-specific antigens which are bound to the non-specific region. So, you perform extensive washing, and then you add a specific antibody to be measured, right? So this antibody is very much specific for this particular antigen, and again you perform washing. So washing actually reduces the non-specific antibody, and then you add enzyme conjugated secondary antibody. So the secondary antibody will recognize the primary antibody, which in turn has recognized the antigen, and add substrate, and then you measure the color. So this is the indirect ELISA, principle of the indirect ELISA. And then we understood about the sandwich ELISA here. As you know the structure of a sandwich. So first you add the antibody that is you use for coating onto the wells, and then you perform extensive washing, and then you add the antigen to be measured. So these antigen are specific for this antibody, so they will bind to that particular antibody, and then again you perform extensive washing to remove extra antigen which is not unbound, and then you add the enzyme conjugated secondary antibody, right? So this secondary antibody is called detection antibody, that is bound with a particular enzyme. So now in presence of an appropriate substrate, this enzyme will convert the substrate. This enzyme will convert the substrate into a specific product, and you can now measure the intensity of this product, and then you have an idea about how much antigen was present in the sample. And finally, the competitive ELISA. This is, as I said, this is one of the most complex type of ELISA that is used in the laboratory, right? Here you incubate antibody with antigens to be measured. So this one is the antigen-antibody complex that you add onto the wells of the microtiter plate, and where these label antibodies are present. So now what happens is that these label antibodies can be replaced by the unlabeled antigen. The unlabeled antigen, this labeled antigen can be displaced by the unlabeled antigen, and here the more unlabeled antigen are present means less amount of radioactivity you will get. Sometimes I mean it's not radioactivity, it is the intensity of the color you get, right? So less color means more antigen is present, and more color means less antigen was present. So that's all about the competitive ELISA. So in this slide basically we summarize three different modifications of the ELISA. The one is indirect ELISA, another is sandwich ELISA, and the final type is the competitive ELISA. Now the thing is, how is ELISA detected? So the detection is important because here, so this one is the capture antibody. If you remember, this one is the capture antibody that actually, as the name indicates, it captures a specific antigen present in the sample and is detected by another antibody that is called detection antibody, and in modern days the detection antibody is conjugated with biotin. Now biotin has a property that it can bind with streptavidin, and one streptavidin can bind with biotin, I guess. So this streptavidin is conjugated with HRP, which is an enzyme horse radish peroxidase. This HRP can conjugate the substrate into a colored product. So this is the detection system. So this detection system allows you to detect the color of the sample, and that in turn says about the amount or the concentration of the antigen present in the sample. Now the final and the most important thing is how the results are interpreted. So ELISA can be quantitative, this can be qualitative, or sometimes this can be non-quantitative. So quantitative ELISA is most commonly and routinely done in the immunology laboratory. Here you see you have got a standard curve, and then in that standard curve, if you put your value that you get using the detection system, and then you can have an easy idea of how much cytokine present in the sample. So that kind of analysis is very important. A proper and accurate interpretation of the results, I mean, you can achieve if you analyze data properly. So nowadays the computers are available, the softwares are available, so you don't have to do anything. Just construct the standard curve and put your value. Then the computer software will tell you how much antigen is present. So you can use that kind of software programs for ELISA. And then sometime it is like qualitative. So in qualitative ELISA, you just get yes or no answer whether that antigen is present or not, right? So that kind of things are routinely done. So what is the take-home message? So here the ELISA is a powerful method not only for general biomedical research but also as a diagnostic tool, because so these days in the COVID pandemic, people also perform ELISA experiment to check the presence of antigens, right? So detecting all types of biological molecules at very low concentrations and quantities. So this is very important, low concentration. Even if the antigen is present at a very low concentration, still you can detect that antigen using ELISA. Although it has its limitations, always it will have some kind of limitations. RIA or ELISA, sometimes called EA, remains an important tool in both clinical and basic research as well as in clinical diagnostics. So as a diagnostic tools, people use ELISA protocol very often, right? So these are my references. So you can study Kubi Immunology 7th or 6th edition and you can get more idea. Also, you can search in internet for ready references of ELISA. I also have an YouTube channel where I frequently upload videos. You can always check my YouTube channel for these kind of videos that are related to a topic. So this is the name of the YouTube channel. So you can just search for the biology made easy. Yes, so ask questions like a child. If you have any questions, I'll take questions now.